Good morning, everybody. I'm Pham from Texas A&M AgriLife Research at the Lubbock Center. For those of you that are growing the crops in the field where the conditions are under 3 digits of the temperature, and especially when the irrigation system is down, for sure in your mind you would come up with an idea like I wish I could have crops in the field that are heat stress tolerant. Like us in Lubbock, it's very hot in the summer. Once summer, the heat unit can accumulate can up to like 3200 to 3400. So those experience growing the crops in the high temperature condition will be much interested in this research evaluation of screening methods for heat stress tolerant in peanut at reproductive stage. Regarding the need for the project, the heat causes adverse effects on crop production and quality, and due to the inconsistent and seasonal limitation when screening for heat tolerance in the field, the necessity of developing a reliable protocol under controlled conditions that allow simultaneous screening of multiple genotypes is needed. Also, the physiological study helped to establish the precise screening techniques necessary to identify traits that are related to plant productivity. There are two objectives in this study. The first one, to examine a set of interrelated physiological measurements under heat stress. The second one is to identify mechanisms of heat stress tolerance in peanut in order to use to screen populations for improved heat stress tolerance, particularly to identify a useful set of physiological measurements that should lead to develop more efficient breeding schemes for heat tolerance in peanut. This study contains in the three main experiments the selected peanut accessions from the U.S. minicore were used for the study. The first experiment, the peanut was exposed to severe heat stress at reproductive stage to identify superior genotypes based on the physiological performance. In this study, we use a method called enhanced respiratory biodemand assay. In the second experiment, the peanut was exposed to the heat stress to study the average number of the flower and the number of fertile pollen. In the third experiment, we studied the method called acquired thermotolerance assay or ATT. Experiment number 1, all of the genotypes used for this study are listed here, and their preference and NBI number were published and are cited in the column right here. In this study, all of the market type are listed in the market type column. The genotype from number 1 to 5 are listed as heat tolerance, and the genotype from 6 to 11 are listed as heat susceptible, and the rest are the check. In this experiment, the control environment facility were used at the USDA, Lubbock, Texas. There are 3 reps for control and stress growing condition conducted in this study. The physiological measurements were obtained are leaf membrane thermal stability and total sugar content. Flowering and pollen stability were conducted in experiment number 2. So how this experiment was studied during the peak flowering stage, the stress treatment plants were transferred to a room chamber for imposing chronic heat stress at 40 degrees C for 7 days. The physiological parameters and average number of flowers under stress and recovery and pollen stability under stress and control were recorded. The third experiment was conducted to study the method called as acquired thermotolerance assay or ATT. It was conducted in detached leaf disc. The method we used was published by Dr. Bird in 1998. So we have known about heat stress, and also we are well aware that multiple measures of heat stress response has been developed. So the question are, what mechanism do these represent? Can a limited number of measures be identified that are enough for screening? These questions can be addressed by the two following methods that we did our study in. Here is how the method enhanced respiratory biodemand assay was conducted. The leaf disc bunches were obtained from heat stress and control plant at dawn during flowering stage, and then the initial chlorophyll fluorescence was determined, and the leaf discs were incubated at 40 degrees C in the dark, and then the fluorescence was monitored every 3 hours for 12 hours. In this method, lower yield of the fluorescence signifies tolerant. The tolerant genotype doesn't have broken heat protection mechanism, so the plant can still respire over the night, and a lot of starch is used up. So in the morning, when the response to fluorescence will be low. This is how the method acquired thermotolerance was conducted. We grew the seedling under intermittent light regime to enhance leaf expansion without significant chlorophyll accumulation. Then we heated the leaf at 38 degrees C for 4 hours to induce the heat protection system. And then we challenge the leaf at 50 degrees C for 30 minutes. And then we keep them in the light for 16 hours to allow chlorophyll production. After that, we measure the chlorophyll accumulation through the spot reading. All of the experimental design and statistical analysis are written in detail in our ongoing manuscript. All of them were conducted in a complete randomized design with 3 replications. We use stats to do the data analysis, and all of the statistical analysis are listed in detail here. Here are the results in our ERBD method. We are seeing that the 3 genotypes COC 41, 50, and 68 are showing a very great potential in heat stress tolerance. Regarding the average number of flowers per day, we are seeing that ICGS 76 and BSS 56 and COC A12 are showing the highest number of the flower under the stress condition, while other like ICGV, Spanko, and Tamban OL are showing the lowest number of the flower under the stress. Pollen stability is one of the factors contributing to use component. In our study, one more data point, we have found that again the genotype ICGS76 show the highest pollen stability, while while the others like COC115 and ICGV have the lowest pollen stability. This is the response of 6 peanut cultivar that are listed on the left side here to the ATT assay. In this experiment, we can see that under the light treatment, all of the leaf punches are still showing green color, while in the ATT treatment, we only see the green color into genotype COC68 and ICGS76. They are still showing green color. In the direct challenge, again, we still see a little bit of the green color in the two genotypes COC68 and ICGS76. So those saying that the two cultivar can serve as a potential to heat tolerance. This is the spat reading data from our study. The blue bar here represents to the light treatment. The red bar here represents to the ATT treatment. Again, from this study, we again found that the ICGS76 showed the highest spat reading under the heat stress condition, along with the COC227, COC68, and COC38, while the other genotype like Spanko, COCA12, and Tambon OL2 showing the lowest bad reading under the heat stress condition. In our result, significant variation was observed among all of the physiological measurements. The correlation analysis showed that the ERBD under stress shows significant positive correlation with relative heat injury. A positive and significant correlation were found between the pollen fertility and flower ratio under the stress. ATT had a significant positive correlation with pollen fertility under stress and negative correlation with the relative heat injury. From our study, we found that under multiple measures, we have found the ICGS76 serve as the high heat stress tolerant. And from our study, we have seen at least 2 mechanisms of heat stress tolerance in peanut. In conclusion, physiological measurement described in this study can be used to discriminate between the heat tolerance and susceptible peanut genotype. And also, the finding in this study, at least 2 mechanisms of heat tolerance, plus the correlation between the ERBD and other measures is very low. So we suggest that heat tolerance can be measured by the ATT, serving as one of the very good measures along with the ERBD. So the source of the tolerant genotype identified in this study can be used to make crosses in order to paramix complementary gene for the same or different response mechanism. To accomplish this study, I would like to thank you, my major advisor, Dr. Mark Burrow, and all of the collaborators and the funding agency. Thank you very much for your listening and have a good day.